Okay, now let's continue the second part of our isolation purification of enzyme. So we've looked at how the enzyme is isolated, and we've seen how we can use ammonium sulfate to precipitate our protein, but that protein is still in the crude state. So how do we purify this protein? Now, we can purify proteins like we said mentioned earlier using some of the characteristics that are specific to such a protein. Okay, and these properties or characteristics that we are looking at is the charge on the protein, the size of the protein, the specificity of the protein or the affinity, and we can also cash in on the polarity of the protein. Okay, so we can actually use these characteristics to isolate one protein from the other in a protein mixture. So now each of these characteristics have a particular procedure that we can use. For instance, if we are trying to isolate or separate the protein based on the charge, then it means we are going to be looking at doing ion exchange chromatography, electrophoresis, or isoelectric focusing. Okay, because all these method or procedures depend on the charge of the protein. In other words, with ions exchange chromatography, you can separate a positively charged protein from the negatively charged protein. Okay, um, even with electrophoresis, you can actually do such separation, and then you can target the isoelectric point of a protein, a particular protein, and if you can focus on that particular pH, you can isolate proteins that that are normally isoelectric at that particular pH. Okay, now we might decide to look at the characteristics and we are considering the size. And in this size, we can look at ultracentrifugation or centrifugation in general or dialysis. Sometimes gel electrophoresis we can separate based on size, and then gel filtration or size exclusion chromatography can be used to separate proteins based on their different sizes. Now, when we look at their specificity, that is proteins that are specific to a particular support. For instance, protein that can bind to ATP, you can always use ATP bound support to separate it from the rest, okay? Because these ones are specific, so you use their affinity to a particular compound which you build in a support to separate such. Now, finally, we can cash in on their polarity, okay, where we might use absorption chromatography, even paper chromatography, reverse phase chromatography, and hydrophobic chromatography. In fact, it will be wise if we can go back to look at some of these methods that we have mentioned, because it's not really part of this course, but we need to know them. Now let's look at chromatography in brief. Now, in chromatography, says a laboratory techniques that we use in separating mixtures. The mixtures are dissolved in a liquid, and we call this liquid the mobile phase. And we say which it carries through the structure holding another material called the stationary phase. Actually, in this course, we are not coming here to actually begin to look at chromatography, the principles and all that, but the principle is that you have the mobile phase and the solid phase. Now, mobile phase is actually your crude protein, okay? Now the solid phase are maybe beads or or gel or resin materials that are poured in with different properties, so that your when you pour in your crude protein, this protein, as they pass through the solid phase, they begin to separate based on the property of the column. Okay, if it is size, they will be separated based on their sizes. It is ion exchange, they will be separated based on the ions that are present on the protein. Okay, so we look at ion exchange. In the ion exchange, we have the cation exchangers and we have the anion exchangers. Now, cation exchangers are used to separate cations. Cations are positively charged. So, cation exchanges themselves are negatively charged. Okay, now the idea is that when we use a negatively charged support, what happens is that positively charged proteins get attached to the support, thereby eluting negatively charged proteins first. So when we want to remove the proteins that are bound to the support, we will pour in a solution that is more positive than our protein. So it will cause the protein to unbound, and then the solution you poured now will now actually, get bound to the support and elute our proteins. In the same way, when you have the um, anion exchange, we say that the anion exchangers are positively charged. Okay, so when you pour your protein, the negatively charged proteins get attached while you elute the positively charged proteins. Okay, so understand it. Anion exchangers, okay, that is the one that we remove the negatively charged, making the negatively charged to come for the support itself will be positively charged, so that negatively charged proteins will get bound, releasing only the positively charged protein. So we have um, the diethylamino 18 as an anion exchanger. It is positively charged, and so on and so forth. So you take your time to study that. Size exclusion, I have been mentioning it over and over again. You pour in your protein into the column. The column is made up of maybe cephadexes with different sizes. Okay, now the cephadexes, they contain beads, and these beads has a kind of holes in it. When you pour in your protein, the protein with smaller size and weight will try to get into the beads, thereby delaying their movement in the column. So in size exclusion, proteins with higher sizes or weight comes out first, while the ones with smaller weight will come out last. Um, chromatography. In affinity chromatography, the support is bound to things probably that have affinity for our protein, like ATP. You use ligands. So when you build them, when you pour your proteins, the protein that have similar affinity for your support will get bound, eluting other proteins first before you wash it with solvent with higher salt concentration. Okay. Electrophoresis, you separate the protein based on charges. I don't want to begin to go in because I believe we have actually done this over and over again. So you can use electrophoresis to separate proteins. You use it to separate RNAs and DNA, nucleic acids, and so on and so forth. Now, one thing we always notice is that during electrophoresis, in most at times, the protein or the RNA is normally mixed with SDS. and this SDS is that's sodium uracil phosphate. Okay, now SDS does two major things to the protein. One, it will help denature the protein. Two, it will confer a negative charge to the protein. Now, when you look at the diagram there, you see that the wells, okay, are placed near the negatively charged. So when you pour in your positive, your protein that is now negatively charged or forced to be negatively charged by the presence of SDS, by the presence of SDS, you will notice that when you put on the protein, by charge differentiation, the charges now, you will notice that the ones that are negative, okay, will move faster because it's running towards the positive charge. And SDS is carrying most of this protein. So you will notice that is why you have different bands showing. On each band you're seeing on the right part is a protein that has been separated. Each band is a protein that has been separated. Okay, so you can use it to separate um, protein. Now, how do we know the weight of the protein of interest? Is a very simple experiment. It means you're going to get proteins of known weight and separate it alongside with your protein of unknown weight. So when you're done, you will actually find the log of the molecular weight of the known proteins and plot it with the distance each of them moved. So when you finish plotting it, you can now trace the distance moved by the protein of interest and you find the antilog. So they are simple things. Um, Isoelectric focusing. In isoelectric focusing, the gel that are used is normally prepared to have a pH gradient. In other words, you prepare it in such a way that the pH of the gel of the column will be of the gel will vary from say 9 to 3. Okay, now remember you will always put your sample near the negative charge. Okay, when you put on the current for the current to pass through, the proteins will be separated. Each of the protein, as they are migrating downwards, okay, any protein that gets to its isoelectric point where the net charge is 0, it will stop moving. Do you understand? And others will continue. So that is why you will see the protein being separate in different bands, and later you can collect from each of the bands. Now we can also separate them based on differential centrifugation, like we've mentioned before now, and so on and so forth. Okay, now if you separate this protein, how do you know the quantity? You can only know the quantity by carrying out a protein test. Okay, and we have different protein tests, and I don't want to go into the principles. They are there in the note. You can also read them up. This is a revision video. Okay, so we have method like the Lowry protein assay. You have the Hattie-Lowry assay, just little modification. um, we have also the modified Lowry method. So you can look at the methods and then know the differences. Okay, you will know the differences between those modifications, what differentiate one from the other. So you have to study that very, very well. Is there in the notes. Now you have the Burette protein assay. Okay, the Baford protein assay. And when you're studying it, always try to know the amount of protein that is needed for this test, because we might decide to ask you which of the methods do you think should be stable for a given amount of protein, or yeah, a specific amount of protein. And why is it that some methods are preferred when we are doing certain kind of experiment? Now the BCA method is there, um, and then, like we explained before, when you have performed an assay for known concentration of proteins, we normally use a BSA, bovine serum and albumin, as a standard protein when we are doing protein tests. Okay, and we generate, for any test you're using, the absorbances for known concentration. So you can actually use it to develop what we call a standard curve or calibration curve. Standard curve or calibration curve. And then from there, you can always determine the concentration of your own protein. Now, during purification, it is always necessary for you to check, you know, each purification test you do, whether your protein is doing better or not. How do I know that the protein is even purified at the end of the day? Okay, so in doing that, you have to carry out what we call purification table. For instance, I want to purify my protein. I did dialysis. After dialysis, I did gel filtration. After my gel filtration, I will determine the place that we have the protein. I did SDS electrophoresis. How do I know that this purification is going on? Yes, in order to purify a protein effectively, the track of the progress must be in check. There are several important quantities that could be measured after each purification step to determine if the purification is working. These several important quantities that can be measured after each purification techniques to determine if the purification, okay, to determine the purification whether it's going on. Okay, so each step you do, it is necessary to do these five things. One is determine the total protein. Okay, this this helps to determine the amount of protein present in the mixture, the whole protein thing, before you begin to remove the the proteins. Now you should determine the enzyme activity. It is always good for you to know the units. For total protein, you determine them in milligram. Enzyme activity, you determine it in micromole per minute, or in international unit, or in what we call kcal. Okay, so you should also determine the specific activity, which is the ratio of the enzyme activity to the total protein. In other words, enzyme activity over total protein will give you specific activity. Okay. You should also determine the yield, and the yield is actually the enzyme activity after a particular purification step divided by the enzyme activity of the crude. And then we determine the purification level. We say we measure how effective each of these purification, each step is in purifying the protein of interest. And how do you get that? Specific activity of that step divided by specific activity of the crude. Okay, now let me just try to um, use the table to explain. Now, supposing you have a crude protein and we want to isolate a specific protein out of the mixture, the following purification steps were measured, and five quantities discussed, and we conduct SDS, okay, on for the protein. We have, after the crude protein, we did SDS. They did sorting out, we did SDS. We applied ion exchange, we did SDS. We applied gel filtration, gel chromatography. We, you know, we did the same SDS, and then when we did affinity chromatography, we also did SDS of the results. And this, the picture by your right shows that. Now, when you look at the table, you notice that the total protein was decreasing as you were purifying, which is normal. Okay, and we also did the enzyme activity after each step. We calculate the enzyme activity. Now, like we say, the specific activity from what we have is a ratio between the enzyme activity of that step divided by the total protein. That will give you the specific activity. So here you have enzyme activity divided by the total protein. So when you do that, you will have all the values for your specific activity. Okay, now one thing that will help you to know whether your purification step is working is not really the yield. I will tell you what the yield actually will show you, but the specific activity and the purification level, once they are on the increase as your purification step is going on, you will know that you are on the right path of purification. Now, what diode does is to show you the amount of your own protein, that protein of interest, that is remaining at each purification step. So if the yield is actually going down, it's giving you an idea that some things are being removed from that protein. So all we try to do is that, okay, for instance, when we finish this work, we have about 35% yield. In other words, that 35% of our protein, of of the total protein, contains the protein of interest. I think it's a very good result. Okay, so the purification table is used to show how purified these enzymes are. I said, as the purification process is going on, the total protein, the enzyme activity, and the yield, um, the total protein and enzyme activity and yield, yeah, we are decreasing, while the specific activity and purification level are increasing. The yield 35% shows that they were able to isolate 35% of our protein in pure form. And that 3,500 here is showing that the protein you isolated is 3,500 times purer than the crude. Okay, so that is just what the so what is normally done in an exam condition is that there's a possibility that the table will be given to you, the total protein will be given to you, the enzyme activities will be given to you, and you will be asked to actually complete the purification table and state whether the purification, the process, the proteins were purified, and give reasons for your answer. As in, if it is purified, you tell us, whether the purification step worked or whether there is an error in the purification steps. Okay, so I think with this revision we have done, if you have question, you know exactly what to do. Our chat groups are still open for you to send in your questions. Good luck.